today I'm just going to touch the very, very the tip of the iceberg, if I say, in the field of genomics. And so this is something I'm doing for well, a large number of years already. So, but still so there's so much to explore. And so there's so many things we can do with this field. So I'm pretty sure I will be really happy. So at the end of the lecture, if you guys at least one of you develop your interest towards studying genomes and the beauty of genomic architecture as well as the genome regulation. Okay, so with that, though, Dr. Kariyawasam gave a good introduction. So other than the academic stuff, I really enjoy the nature and photography. And so that is something I do when I'm not doing science. And one day I would like to retire and do it full time, hopefully. I don't know with the Sri Lankan, the economy, whether it's possible, but probably, 2048, we are going to be a developed country. So therefore, so we might be able to, I might be able to do that with myself and my spouse. And if you look here, there's I was trying to catch very tiny common kingfisher. Anyway, and this is me. Okay, so these are different the contact information. I have my own website, and then I have my own YouTube channel, and then my photographic stuff. If you guys have time, feel free to visit them and take a look at it, and then maybe it will help you to reduce your stress. And when it comes to the genome stuff, very quickly, I will tell you what I'm doing. I study how plants respond to stresses. So what we do is we basically look at the whole transcriptome level changes, especially in crops, with the objective of developing crops. So those are future proof. So that means they can withstand all this the climate change we are talking about and all the other the environmental disaster environmental assaults some say. And so that's one of the things I'm doing in addition to that, like I said. So this field really amazed me. So therefore I'm not restricted to plants. So one of Dr. Kariwasam's student did the mushroom project. I helped that, and then we were working with the Sri Lankan leopard. And nowadays we sequence three genomes belongs to Sri Lankan bees, especially Apis mellifera, serrana, and dosata. So I already received my sequences, and so I'm waiting to assemble their genomes and then see what are the differences when it comes to our bees versus the bees found in the other countries, Asia as well as the Western country where they have an Apis mellifera. And also we did a couple of aphid projects and then some human disease related projects. So there are so many projects we're doing. So that's the beauty of it. If you study genomes, so you're not restricted to one species. So any living thing, they contain DNA, RNA, and protein. So therefore, you can study. Okay, so when we start talking about our genome, or not necessarily our genome, all the genomes found in this, the Mother Earth. So there are so much to explore. But now the question is where we started. Actually, if you especially think about our genome, so that is one of the biggest project launched in 90s, and it is launched in 1990. And I don't know whether you will believe the estimated cost that was spent on this human genome project was 2.7 billion US dollars. And it took more than 13 years to complete this project. And so this project involved in a kind of consortia. So where the scientists from three continents, so they basically came together, and in 20 collaborative sites, the research labs. So they were and both private and public. So they were doing this sequencing. Okay, so imagine 2.7 billion in 1990, and Surprisingly, now it's like, in 2022, like 2 years back. So now we have the ability to sequence the human genome just by spending $100. So that's how things change, like from 2.7 billion to $100. So imagine, like, so now if you have like, $100, you can get your genome sequence. So that was the objective, and we already achieved it. Maybe in the future, it will be even lesser. So that means we can do so many things. We can do personalized genome related, like personalized medical care. So where, so the when you go and visit your doctor, so he will like basically diagnose your disease, and then so you already have your genome in their like database. So where he will basically prescribe you or like a specific drug that specifically target for you. Okay, so that's the level we can go for. I mean, already there are some the personalized genomic therapies, but it's like little expensive. But in the near future, we would be able to harness the power of genomics, especially when I mean, like if you think about our health. And then this genome was published in the special, I mean, like the in Science, and you see like how many people involved in this project, and this is only a part of the list, and because it's a consortium, there's like so many other people. So now you're thinking, okay, I study, like me and you, like all of us study plants, not human. So what about the plant genomes? When you think about the plant genomes, the Arabidopsis is the first genome to be sequenced in 2000, and followed by the rice genome. So there were like two versions of rice, like cultivars, what we call as japonica and indica. So they were sequenced in 2004. Now you're probably thinking, what is superior, the Arabidopsis or the rice? But if you look at the sequence quality, I mean, I'm talking about the past. And at that point, rice, it had like a very, very nice linkage map. Maybe that's something you learn. So therefore, like, it was like, very easy to like put sequencing detail into the linkage detail. So that way, the rice map was like the resolution was, of the rice map was relatively higher than the Arabidopsis. Okay, so now, like the major species that was a sequence in like early days. If you think about the timeline, so everything started like 2000 afterwards. So the last 2 decades, so we are like basically like show like two different types of progress. So we will start in the first decade. The first decade is called the notice, and it's this is not a joke. Actually, it is this decade from 2000 to 2010 was called notice, and so during this decade, so the objective was to provide high quality reference genome for model plants and crops. Here we are specifically focusing on plants, and then if you think about like this genome sequencing and assembly, so they were like, it was like, a major undertake at that time. And during this 10 years, only 26 genomes were published. Okay. And all of these genomes were published in Science and Nature. Okay. So, the Science and Nature, like, I dream to publish things in Science and Nature because those are like two, like, not the highest rated, but like, two of the best journals we can go for, like as a molecular biologist or like a genome scientist. And if you think about these papers, so they had like the same thing, like we followed this procedure, and then this is the genome. Okay, so even though that is the amount of data they had, so they were considered as significant breakthroughs. Okay, so and then the beauty of it is, so it provided the foundation for the subsequent research related to genomic studies. Okay, so therefore, so we can't like even though only 26 genomes were sequenced during this the notice decade, so but the contribution given by this decade is like it's amazing, and it's like basically you can't like evaluate because that important it was for the the subsequent decade. The next decade, from 2011 to 2020, we call it as the tens. And there, what happened was with the advancement of sequencing technology, that is something we are going to talk about today. So the number of sequence plant genome went up by more than 50-fold. And now, if you think about, okay, so now there are so many things coming out, and what about the publication or publishing? And what happened was the high impact journals, they become very interested in assessing the impact of these genomes, and then the biology of the plant. Okay. And that leads to the genome based discoveries. So that means, so those were not only, okay, so this is the genome, we sequence it in this method. Rather, now journals are asking for a lot of interpretation, how you link it with other biological aspects of the species of interest. Okay. So that is the second decade. Now we are living in the third decade. Okay, so let's see, how much cool things we can do during this decade. I mean, myself and you guys, because you guys will also, and at least some of you will be working with genomes, and then you're going to contribute to this to this field. And if you think about the timeline, just to get an idea, so this is basically the timeline, as you can see here. So the initially there were very very small organisms, and their genomes were sequenced. And you can see the time in between each and every genome. Initially, it is sometimes many years, but so after you cross the 2000, the notice decade, see what is going to happen. So basically now the distance is becoming short and short. So that means more genomes are coming out from sequencing just one genome. We went for 1,000 genomes. So imagine the leap the scientists took with the help of these new advanced technologies. Okay, so therefore so I will share the slides afterwards, so you guys can have a look at it very closely. Now we are talking about not only 1,000 genomes. Now we are talking about 1,000 genome, and then so I will there's a surprising the thing or initiative. So that I will will explain towards the end of the lecture. Okay, so we talk about the how genome level evolution happened during this the last 20 or 24 years. What about the sequencing technologies? So that is something we need to closely look at. So when you think about these the sequencing technologies, as you were aware, so the first generation sequencing was very costly, and then there were many limitations associated with it. Okay, so first generation sequencing, it was started with Sanger. Sanger introduced both the genome sequencing, DNA sequencing, as well as the protein sequencing, and so he contributed a lot to the advancement of this. Even though now we consider first generation sequencing is it's kind of slow thing, but in reality, it contributed a lot. And Sanger sequencing, initially it was the gels and then the manual gel-based method. And then 1990s, it was advanced to to make it automated. So that way you can do more, you can have more throughput towards the end. And so therefore, the capillary-based sequencing of Sanger sequencing was introduced, and that those things led to the Human Genome Project, and then the Christ Genome and Arabidopsis Genome. Likewise, in 2000s, what happened was, so the next generation sequencing was introduced. So that next generation sequencing, we divide into two groups: second generation and third generation. The second generation sequencing includes Illumina, Roche, and Ion Torrent. So these ones are very high throughput. Okay, we will look at each and every technology in a bit. And the thing is, now instead of making, remember, I think all of you are familiar with Sanger sequencing. So if you're really, really lucky, you will get like a very nice 700 base pair read from like, one round of sequencing. But the moment you jump into Illumina, what you're getting is like millions of short reads. So, so therefore, like, instead of like, getting just one sequence, now you're getting millions of reads. So therefore, like, so you can have like, more throughput. And then the second generation, so one limitation was most of the second generation sequencing technology, they were like basically amplifying small areas, or what we call as like the small reads. So therefore, uh, it's some like now you have like it's like uh, working with a puzzle with 10,000 pieces. So imagine you're like making a puzzle with 10,000 pieces. It's going to be a nightmare, right? I mean, it's fun, but it's going to be like a nightmare. So the second generation was like that. And then that leads to like, some issues. And then as a result, the couple of other companies, they developed third generation sequencing. So that includes PacBio and then Oxford Nanopore. So the beauty of these technologies is they can do long reads instead of like 600 base pairs or something like that. So now you can do like, mega bases, okay, or kilo bases or mega bases. So that way, so you have long reads, and then so now it's easy to assemble, right? Now you have like, a puzzle with let's say 500 pieces. That means your puzzle pieces are big compared to like, 10,000 size puzzle. So that means you can easily put together and then the assemble the genome. And then there were like, even more new technologies were introduced. Genesis was one, and then the MGI was another one. But unfortunately, last year, Genesis, the company, it went bankrupt. So therefore, I don't think any company is working on that technology. But MGI is something now everyone is working with, especially like if you sequence things from a um, is the BGI, Beijing Genome Institute. So they use this DNA like they call as balls or nanoballs in order to do the sequencing. So that is again like advanced system. So we will discuss or we will look at videos for like in each and every technology stated here, except for the Genepsis, and then you will understand the principle behind it, and then so there are instances where we can combine not one technology but multiple technologies to have a high resolution database. Okay. So far, do you have any questions? If not, I'm going to proceed. So I'll give you like 30 seconds, and if I don't hear anything, so I will proceed to the next sections. So here we talk about again small and large, Okay, so I was thinking like, our students, they never ask questions. So I think the SGP is not so different. Okay, so anyway, we're moving forward. So we are going to talk about the smallest and largest genome. I don't know how many of you follow American politics. I mean, I don't know why, but like, it's like my second home. So therefore, I follow their politics like very closely. And if you guys get a chance, go to YouTube and search for like, the Obama's latest Democratic National Convention talk, and then you will understand uh, so the context of this the picture. Okay. So anyway, moving it's something to do with like Trump and then the like he was referring to Trump. Okay. So anyway, uh, so you guys have any idea about the smallest plant genome? Actually, if you think about the smallest plant genome, it's so far the smallest is the Genelisa tuberosa, and then the genome of this plant is only 61 megabases. Okay, so very tiny. And if you compare it with the largest genome, so they are like the until recently the largest genome was the Paris Japonica. It was around 150 gigabases. Okay, so the smallest is 61, and then the largest is 150 gigabases until recently. But I think your sir is like very fond of ferns, and like it or not, the largest genome is coming from a fern, and it's very difficult to pronounce the name of this. I'm sorry if I pronounce it wrong. It's called Demis.
Visit cherries. It's tea silent.
Oh, yeah. See, I
Yeah,
was waiting till you.
yeah. I
So,
was thinking like because the Ophioglossum reticulatum was like 2n equals like 1260 chromosomes. It
uh,
was there, and then this recent paper from Nature, I think, yeah. So they published this massive cherries. So
yes.
it's the same family, and they are very sister. So it's very surprising to see this came out very recently, Professor. Yeah, it's
Yeah.
tea silence, right? Silent.
Yeah. Okay. Yeah. So I was so worried that,
Yeah, and also here it's like T silent and then P silent. It's Messiteris.
massive terrace. Okay.
Yes,
So
yes. Thank
yeah,
you.
actually, I try look for like a pronunciation in YouTube as well.
Uh-huh.
So, but like it was too short of time for me to memorize the actual pronunciation.
Very
So
welcome.
yeah. So anyway, so the size of this is 160 gigabases, and then if you basically take the genome and if you spread it inside one cell, it's like having 100-meter long rope inside of it. So imagine the amazing how it is compacted into one cell. So I mean, so it's amazing. I mean, it always amazed me when we think about this the how these genomes are assembled inside a single cell. Okay, so and so why, I mean, because of the advancement of genomic sciences, we know this kind of cool information. So therefore, again, it's something a kind of a carrot for you to follow the genome sciences. Okay, so moving forward, so I mean, if you think about the things happens and all the key things circulating around the genome world. So these are the things we are talking about, talking in last 20 25 years. Okay, starting from tDNA insertion lines to expression profiles to 1,000 genomes to the databases like TEA and BA, and then StarSeq, and then different sequencing techniques like DAPSeq, KTAC, so there are so many keywords floating around. So when you start studying this, so these words will appear. So don't get scared, and you can always find resources to find a very detailed information about these techniques, and I'm pretty sure. So the probably all of you are so interested in genome editing. So these days CRISPR-Cas is one of the hottest topics circulated around, where you can do uh, changes in the plant and animal genomes, where you can use it to cure diseases and make crops without introducing any transgenic or transgenes from other species, because some people are so afraid of transgenic plants. And I mean, there's no reason for it, but unfortunately the social media and other the pseudoscience, the scientists they basically create fear. So I mean, that is what happened to Sri Lanka until, right, I mean, so far we can't do any genome studies, the transgenic study, because of one or two pseudoscientists, because we don't have the biosafety act out. It is stuck in the legal draftsman for last 12 to 14 years. Okay, so that is a story for another day. I'm not going to talk about it at this point, but if you're interested, you guys can always send me an email and ask me. Okay. So moving forward, and if you think about the two first plant genomes sequence and how they were used. by different field. So this is basically a diagram showing how different uh, journals and different researchers cited this genome sequencing of Arabidopsis and rice. You can see it is not only restricted to genomic, but so they're cited in agronomy to genetics to biochemistry to forestry to microbiology. So therefore, whatever the genomic study we are doing, they will be applicable for a vast range of uh, fields. Even the not only plants but also the human uh, uh, human related studies. They also refer to plant genomes in many instances. Okay, so we are going to look at the latest, until last March, how the genome sequencing are deposited, and then what are the numbers. If you look at the genomes deposited in NCBI in this last March until last March, you can see number wise there are more animal genome sequence compared to plants. So if you rank them, the number one is animal, and then number two is fungus, and number three is plants. You can see there are 1,482 genomes. Those are completely sequenced or complete genomes available in the NCBI. And if you look further, so you can see if you look at different the groups of plants, you can see so the angiosperms. So they're almost 90% of the genome represent the angiosperms, and then the number two is chlorophyta, and then likewise as you can see, so there's always a bias because depending on the model organisms and depending on the active research areas, so we tend to study our favorite species. So that's why there's the diversity, or there's a difference in the number of sequences deposited in the NCBI. Okay, so and then when you think about the genome, so whatever the genome that is deposited can be uh, just the set of sequences. Okay, so they are not basically divided into chromosomes. Okay, so that means they are incomplete genomes, as you can see in this pink color, I guess. Yeah, pink color. And then the blue color represent the chromosome or the complete genomes deposited in the NCBI. And then this beige color give the cumulative number of sequencing genome. And then you can see the number of species sequenced during this time. As you can see here, early days, you have the majority of non-chromosome. Because remember, I told you that it is difficult to sequence things from end to end, from one end of the chromosome to the other end, because of the limitations associated with the sequencing technology. But with the help of third generation sequencing, now you can see in recent years, the amount of the complete chromosome or the complete genomes at chromosome level, so the number went up because of this the long range sequencing. And if you look at this the sidebar, you can see the genome sequence, the key species, those are sequenced, and the timeline of their sequencing. Okay, so likewise, so the take home message is now there's an basically explosion or an exponential increase in the number of genomes deposited in the NCBI with the advancement of this the technology. And the most interesting thing is, so now we have genome, so therefore now we can get an idea about the genome sizes. And in some groups, you can see there the genome sizes, the range is huge. Okay, so compared to some other groups. And if you look at this, the right side chart, you can see the number of species sequence, and then the left-hand panel, it shows you the genome size. So therefore, when you think about plants, so they have like the remarkable diversity, not only like at the appearance, outside the appearance, but also inside their genomes. And if you look at the different sequencing technologies people use in order to do this, uh, initially until like 2017, 18, so they were like the either Sanger or the second generation sequencing technologies like Illumina or Wine Torrent, also likewise. But now all these green dots, they represent the third generation long read sequencing technologies. So therefore, that's why you have more sequencing, and then because of that, you can see that the size of contigs. So when you assemble genomes, so you make contigs. From contigs, you can make scaffold you turn into chromosomes. So the N50 of a contig is like if you take the 50%, more than 50%, if you take like 50 point, so then if you take all the other contigs you have, they have more than this specific size. That's the N50, and then you have L50. Um, so the N50 is now it's going up because of this long range sequence. That means you have long fragments of genome that can be easily assembled into higher levels like chromosome level. And then probably you're thinking, what about the protein coding genes? So let me ask you a question. Uh, so you can use the chat window. So can you guys tell me how many genes in a human cell, or as humans, how many genes we have? So you guys can use the chat window and see okay, so you need to promise me that you're not going to Google it. So don't Google it. So just I mean it doesn't matter whether it's correct or not. So just guess and then put how many genes you have in your in a human uh, in humans. Yeah, so I'll give you 30 seconds. It's enough, right? Yeah. I guess all of you are like sleeping or something because I don't see any response so far.
Students, you can use the chat window and put your just guess and put your response. There's not nobody to penalize you, right?
Or else we can take 50% of their marks from the examination. Right. Okay. So there's one answer. Okay. Two. Okay. So I'll wait till I get like five answers and then I will move forward. So far two. So waiting for like another three. Okay, cool. I have like five answers, uh, ranging from 1,000 to 30,000, 20,000. Okay, so from 1,000 to 20,000. And so you won't believe, initially, so you have 3.1 billion bases, in genome. So then, people, they were thinking, okay, so we have 1,000 genes, because each amino acid, so you have to have three nucleotides, no? So therefore, people thought it's 100,000. But in reality, what they realize is once they started the sequencing and doing the genetic study, it's only so far. I mean, based on whatever the evidence we have, we think it's around 19,000 to 23,000 range, and not more than that. So with time, the number reduced, and we also realized 85% of our DNA scores junk, but it's not really junk, but junk DNA. So therefore, only 19,000 to 23,000 genes in our genome. And when you look at the plant genome, you can see there, based on whatever the species we have right now, so the mean number of protein coding gene is around 34,071. And if you look at the distribution, if you think about different plant clades, you can see. So most of the clades, even though the genome size increases, there is no a significant increase in the number of protein coding genes. Okay, so that is something amazes scientists when it comes to studying protein coding genes. Okay, so with that, what we are going to do is we are going to look at different sequencing processes. I'm not going to talk about the first generation or the Sanger sequencing, but rather I will stick to next generation that includes both the second generation as well as the second, third, and future generation, all three. Okay, so when you think about before that, I forgot to mention uh, the estimated. I mean, if you think about the number of green plant species around found in the our Earth, it's around 450,000 to 500,000. And then so we have 1,482 good quality genome sequences. So that means so far we only sequence around 0.3% of the plant species available in the world. So therefore, a long way to go. So don't worry. So therefore, even though you see exponential growth, don't worry. You will get your chance to sequence your favorite plant genome without much competition, because there are another 400 to 500,000 species available for sequencing. Okay, so moving forward, when you look at these different sequencing technologies, even though the specific steps are a little bit different, so they all the sequencing technologies they need couple of common steps. Okay, so you have your sample, and then what you're going to do is basically you need to shear it into fragments, and then you will do the end repair uh, to make a blunt end fragments, and optionally you can do a tailing in order to increase the ligation efficiency. And then what you're going to do is you're going to put adapters. You will see why we need adapters. So we basically put adapters and we ligate them into our fragments, and this is what we call as PCR library. And sometimes so we can move forward with these PCR-free libraries, but some technologies you need more copies. So therefore, you go for an amplification step. So where you amplify your sequences in order to do the sequencing. So therefore, these are the very basic steps involved in next generation sequencing technologies. Okay, so simple as that. So to show you another diagram in more illustrative manner, so please take a closer look at it. You have your DNA sample, and as I mentioned earlier, so you basically shear it into fragments, and then you need to repair the ends Okay, and also you add A's. So in order to increase the ligation, and then you do the adapter ligation, and then if required, you can add barcodes. You will see why we need barcodes. Because if you do multiplex, you need barcodes to separate. Okay, so this sample is coming from this plant. This plant is coming. I mean, this sequence is coming from this plant. Likewise, and also the amplification. Remember, I told you some technologies you need more copies to do the sequencing. And then if you follow the Illumina, so you have like you use Illumina library, and then basically you can see you have the adapters, and then you have the barcodes, and then you have the linker. You will see why you need a linker. So when I show you, I mean, I'll show you a video. And then for PacBio, so they use a different technology, which what we call as the smart bell. And like you can see like the one the sequencing part is basically like a fragment with like two loops, and then Oxford Nanopore. So they use like protein motor technology. So therefore like you have linear one, and then you put a like barcode. So because then you can do multiplexing. And now, so we have like a more advanced way to do it. So just by using like transposase. So thereby we can like go from direct DNA sample to adapter ligation in one step rather than going with the shearing and then the end repair. Okay, so therefore that basically saves more time. Okay, so when we look at like two most common and most highly used sequencing technologies, so this is the what you call the what is happening inside the machine. Okay, so when you go and look at like Illumina HiSeq or like uh, MySeq or NovaSeq, so this is what is happening inside that machine. And then when it comes to PacBio, so they have different chemistry. And then when it comes to Nanopore, so you have different technology. So the Illumina, what we call as the sequence by synthesis system. So where you have the flow cell, where like you have like basically linkers. That's where our sequencing fragments are going to bind, and then we use the polymerase to amplify it, and then you basically using a laser, you look at the fluorescent when it incorporates bases to the fragment, and then so basically like the detector can read whatever the specific sequence based on their fluorescent chemistry. And when it comes to the smart sequencing, instead of like using this kind of technology, so they are like basically focusing on single molecules or the single fragments, and then so they are basically they follow the smart sequencing method in order to do the amplification. Again, this one is fluorescent based. Every time a base is incorporated, it can be detected by the trace, or the trace can be detected by the detector, and then it will be converted into so basically ACTG sequence style. And when it comes to nanopore, that's they follow a completely different system. They are not basically relying on the polymerase and linkers. Instead, so they found this the the molecule. So this the motor molecule. If the beauty of it is, so it has like a small hole inside, and it is around 1 nanometer, and so that is ideal size to like basically take a single strand inside that pore, and then they create an artificial membrane where they maintain a current, like the voltage gradient. So what happened is you already know DNA is negatively charged. So therefore, like if you have a positive gradient on the other side of the membrane, so the molecule is going to move. And then every time it like basically comes out, what is going to happen is there will be a like a photovoltaic change, so that the change in the electricity can be detected because it's specific for ACTG. So, and then, so that way you can basically get your ACTG or the sequence order with this technology. And nanopore is like, really cool because so the size of a nanopore sequence machine, can vary from like, like small cassette, let me see, from like, something like this to like, one big, like, freezer size machine. They have Minion and then they have Prometheus. And so these different machines, they have like a different throughput. The small one, so you basically like, put it inside your backpack because you need a laptop and then the flows it. And then the machine, you take it to the field. That's what they did in 2014. There was an Ebola outbreak in Africa. So the scientists, because there you don't have enough electricity, you can't carry like, like big machines, and they can't afford it. So instead, what they did is like they basically took this the minion to the field, and then they did the sequencing like on site. Okay, so that is one of the advantage of Genopo sequencing technology. Okay, so the next we are going to look at a couple of videos. They basically like in very simple manner they explain each and every technology. Okay, so we will start with Illumina. So let's see. If you please let me know if you can't hear the voice. I shared the sound, so therefore should work fine.
Sample preparation begins with extracted and purified DNA. The first step in Nextera sample preparation is tagmentation. During tagmentation, transposons simultaneously fragment and tag the input DNA with adapters. Once the adapters have been ligated, reduced cycle amplification adds additional motifs, such as the sequencing primer binding sites, indices, and regions that are complementary to the flow cell oligos. Clustering is a process wherein each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with a lawn composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process, the strand folds over and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, 4 fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determine the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the Index 1 read primer is introduced and hybridized to the template. The read is generated, similar to the first read. After completion of the index read, the read product is washed off and the 3' end of the template is deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as Index 1. Index 2 read product is washed off at the completion of this step. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends blocked. The original forward strand is cleaved off and washed away, leaving the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is washed away. This entire process generates billions of reads, representing all the fragments. Sequences from pooled sample libraries are separated based on the unique indices introduced during the sample preparation. For each sample, reads with similar stretches of base calls are locally clustered. Forward and reverse reads are paired, creating contiguous sequences. These contiguous sequences are aligned back to the reference genome for variant identification. The paired end information is used to resolve ambiguous alignments.
Okay, so that is the Illumina technology, or the sequencing by synthesis. And we will watch another video, and then probably we can have 5 minutes break, so you guys can have get up and then have a cup of water or tea and come back. So we will watch this one and then have a break. So this is PacBio.
Introducing the PacBio Sequel II system, powered by single molecule, real-time, or smart sequencing technology. Here's how smart sequencing works. First, from any sample type, ranging from viruses to vertebrates, DNA or RNA is isolated. Next, a SMRTbell library is created by ligating adapters to double-stranded DNA, creating a circular template. Primer and polymerase are added to the library that is placed on the instrument for sequencing. At the core of smart sequencing is the SMRT cell, which contains millions of tiny wells called zero-mode waveguides, or ZMWs. A single molecule of DNA is immobilized in the ZMWs, and as the polymerase incorporates labeled nucleotides, light is emitted. With this approach, nucleotide incorporation is measured in real time. With the Sequel II system, you can optimize your results with 2 sequencing modes. Use circular consensus sequencing mode to produce highly accurate long reads, known as hi-fi reads. Or use the continuous long read sequencing mode to generate the longest possible reads.
So far, we looked at the Illumina and then PacBio. So let's look at the nanopore sequencing. And as I mentioned to you earlier, this is one of the coolest technologies when it comes to the sequencing. So look how small it is, the medium. And I forgot to mention that this was used even like in the ISS, the International Space Station. So there, basically, they took the one of the minion and did the sequencing. So therefore, so it basically went from Africa to the outer space, or space boundary, um, to sequence things both inside as well as the outside. Okay, so next, we are going to look at the future generation. Remember, I told you there are two different companies, and one went bankrupt. So this is the what we call as the DNA ball, nanoball technology. So this is practiced by especially by the Beijing Genomic Institute, and they use a different chemistry to do that, and it's very similar to Illumina, but not exactly same, because they have their own pattern for their technology, and Illumina, they have their own. So let's look at it. Okay, so that is the DNA nanoball. So now you might be thinking, okay, so we have one set coming as small reads, another set coming as a long read. So what is the optimal situation? So and people these companies also thought about it, and then they basically came up with the thing called one example is Hi-Fi Seek. So you will see how you can combine different technologies in order to achieve more higher resolution.
As scientists, we're fascinated by the beautiful complexity of our world. I mean, that's why we became scientists. But the more we learn, the more we realize that a lot of the answers we're looking for, the good stuff, are locked away inside the complex genome of an endangered species, as part of the genetic variants causing a rare disease, hidden in a microbe with its potential to help or harm us, or within one of thousands of other mysteries yet to be uncovered. Unlocking those secrets takes more than just brain power. It also requires the right technology. Unfortunately, a lot of sequencing technologies require compromise. To get a complete picture of a genome, you must assemble the reads. Short-read sequencing provides high accuracy, but only small snippets of data, giving an incomplete picture. Or traditional long-read sequencing that offers a bigger picture, but lacks accuracy, so it's difficult to tell the difference between true biological variation and errors. But highly accurate long-read sequencing, or Hi-Fi sequencing, is changing everything, combining the best parts of short reads and long reads into a single, easy-to-use technology, creating a whole new paradigm for DNA sequencing. Hi-Fi sequencing's read lengths are really long, hundreds of times the length of short reads, and achieve greater than 99.9% accuracy, on par with short reads and Sanger sequencing. Easy library preparation and faster compute time mean quicker results, to keep both the biologists and the bioinformaticians happy, and to keep costs down as well. Most importantly, however, Hi-Fi sequencing can enable us to tackle the biggest and most complex genomes, no matter what field we're exploring. conservation and agricultural advancements, human health and disease research, microbiology and infectious disease, and beyond. To empower scientists like us to strive for the breakthroughs that have previously been just out of reach. Hi-Fi sequencing from PacBio. Unlock your next great discovery.
Okay, so see, you can combine good things from both long read and short read sequencing and get very more than 99.9% accurate outcome. So now we are wondering how we can put everything into one picture. So what we can do is basically you can use the short reads the second generation sequencing technologies, and then you can combine it with the third generation sequencing technologies where you have long reads, and you can combine short reads versus long reads, and then make bigger context, and then as a result you can make bigger scaffolds, and so that basically enhance the quality of the genome. And so but one thing you might be thinking remember we have 23 pairs of chromosomes, and we can put things right next to each other, but how we can determine, okay, so these ones are located very close to each other, because when you think about the genome inside the cell, so it's very tightly compact. So therefore you have to have a good idea about things around us. To give you an example, I think you learn about the promoter. So you have promoter, cis elements, and then the enhancers. Sometimes enhancers located far away, but but they're somehow, because of the folding, so they are very close to your favorite gene. So therefore those things we need to understand. So to do that, we can do what we call as high C scaffolding. So where not only you get from the short and long read sequencing, you get very high resolution scaffold, but also you can put them in the correct orientation and correct vicinity using this what we call as high C scaffolding. So the next video is going to show you a very, very brief introduction about the high C scaffolding. So let's look at it.
Proximal high C. We start with intact tissue from plants, animals, and human samples or cultured cells. Chromatin is packaged into three-dimensional structures that retain a relationship between genomic and physical distance. Sequences that are closer on the same chromosome are also closer in physical space. Our method exploits this relationship between linkage and proximity to enable whole chromosome scaffolding and phasing of genomes. The DNA in the sample is cross-linked in vivo to fix DNA sequences present inside the same cell. Cross-linking traps sequence interactions across the entire genome and between different chromosomes. Cross-linked DNA is fragmented with endonucleases. Fragmented loci are then biotinylated and ligated, creating chimeric junctions between adjacent sequences. This process is called proximity ligation. The more often two sequences are joined together, the closer these two sequences are in genomic space. Biotinylated junctions are purified and subjected to paired-end sequencing. The proximity ligation reads are then mapped onto a draft assembly. Proximity information is used to assign contigs to chromosomes and order and orient them along chromosome scale scaffolds. This results in fully scaffolded chromosomes of virtually any size. This process also detects structural variation and corrects assembly misjoins, as well as maps the three-dimensional conformation of chromatin within a population of cells. Bring the power of high C into your lab today. Learn more by visiting phasegenomics.com.
Okay, so this is how we basically look at how our genome is assembled arranged inside the cell, so that we can get a much better understanding about the genome dynamics. especially when it comes to functional analysis. This is extremely important, especially like I study how cis elements enhances regulate the gene expression. So for this is like golden technology that can be used in order to identify how like a very distal enhances and the promote the cis elements close to the gene is regulating the gene expression. Okay, so this everything looks very simple, but actually, if you like try to assemble your favorite genome, so there's a process. So, like as a Sri Lankan scientist, like one of the major limitations you might face is the computational power, because making a sequence, genome sequence, is easy than analyzing it. Because nowadays, I mean, even though like in Sri Lanka there are like a couple of I6 and couple of My6, so personally we send samples outside and we don't worry about the sequencing part. It is done by a different company. For us, it's like usually Macrogen, and this then send us the sequencing files. So usually, like the when we received our the base genomes, so each genome, so there were around 8 gigabases. Okay, so therefore, like in our files, so they were like at least the FASTQ files, they were around 6 GB as far as I remember. So therefore, like 6 times 4. So that is the amount of GB we downloaded from their link. So and that is what we use. You might think like 6 GB is nothing to work with, but when we do assembly, the size like it's going to increase exponentially. Because when you go from FASTQ to SAM, sometimes SAM file is like 40 gigabytes. So then you need space, and also you have to have a memory. Like the computer I'm using right now, it has 128 gigabytes of RAM. Still, I can't use it for a smooth genome annotation. For that, I'll show you in a bit. So where I had to use a supercomputer found in because I am a remote collaborator in Texas A&M University. So they give me the computer access. So therefore I use their supercomputer in order to analyze my data. But sometimes like initial assembly, even with they have two thousand 64 gigabytes of RAM, and then you can have up to 8 cores of CPU, and still sometimes it take a couple of days. Okay, so therefore computational power is going to be the limitation, but there are some free tools. I will introduce one tool towards the end of the lecture, so that is something you can very easily use in order to assemble your genome. It will take some time, but it's better than nothing. Okay, so if you look at the whole genome assembly pipeline, it is something like this. Okay, it looks complicated, but once you start doing it, you find it it's not that difficult thing, because if when you basically optimize your pipeline, then so whatever the new genomes coming up, so you basically input it and then you run the same script to get the output. So therefore, getting everything in order at the first instance is the tough task. Thereafter, it's not that difficult. Okay, so once you receive the sequences, so it can be short reads or it can be hybrid, that includes both short and long reads. And long reads, so and this is kind of a decision tree. So you pick the ideal coverage for your genome, and then you send your samples, and then so you're going to get whatever these different types of reads. And then if you don't have a reference genome, you do de novo de novo assembly. But if you know a reference genome or very closely related reference genome, so you can do reference guided genome assembly. So which takes less, I say, computer power compared to the de novo assembly, because de novo assembly, you have the computer, it has no idea the orientation, it's just like a jumbled puzzle. So no picture to show, okay, so this is the outcome of the puzzle. Okay, so therefore it's it's more it requires more computational power compared to the reference guided genome assembly. And then, so once you have your assembly, you are going to basically polish it and make it beautiful using different tools. Like in some examples are given here, but there are so many different open source like free tools you can use. And then once you polish it, so then you can basically refine it, and you can confine it. And so you look at different parameters like context size or the N5, N50, and then the boost score to see the amount of coverage. And it is good for chromosome assembly. You're going to move forward. If not, you will basically deposit as a draft assembly. And then the chromosome level assembly can be done using different tools. And I already introduced you the IC. So where you can have the chromosome scaffolding, and then so then next step is basically you annotate it, and then so you can make a genome browser. I don't know whether you're familiar with Ensemble Genomes or Gamine or even the NCBI. They have their own genome browsers where you can easily visualize it. And then so once you deposit it, and then so the other people will also sequence your favorite genome, and then you can do curation and refine it and then make it even more perfect. Okay, so this is how you basically assemble genome, and go from a draft genome to a complete genome. And sometimes you have complete plus an enhanced genome. If you think about the human genome, only last year they finished the refinement. So now only we can say we have sequences from end to end for each and every chromosome. Because earlier, even though the human genome came out in 2000, actually the telomeres, the centromeres were not sequenced. Okay, so they were completed very recently, and also the telomeres. Okay, so and when you think about our plant species, you can see the how different groups of plants are sequenced, and you can see that the first one, this pink color, it indicates domesticated plants, and the purple one is the cultivated plants, and then blue shows you the feral ones and natural community, wild relatives and wild. You can see. So most the big countries, so they have a lot of both the domesticated and then the wild plants sequence in addition to the cultivated plants, but it varies. You can see US and then the Europe, and then including UK, and then China and Japan and Australia for a certain extent. They are because these three are the pioneers when it comes to genome sequencing. And you can see the number of, based on the size of the circle, you can see the number of assemblies came from these countries. And now we're wondering, okay, so these countries, they have this much of species richness or not? Okay, so when you look at the genome sequence, and this gray color indicates the native species, and then the white non-native, and then the black is the native species sequence of continent. So you get what I'm saying, right? If you take North America, you can see, so the let's say around 8 genomes were sequenced belongs to that specific continent. But if you look at this, the white color one, so that includes non-native species. So that means, so they sequence the genomes coming from different countries. And it is true for Europe as well. But if you look at the Asia, it is quite the opposite. Okay, so there is a disparity when it comes to the sequencing, because it's very clear, right? Because so they have more money, we have less money, so therefore we tend to collaborate and then do the sequencing elsewhere. Okay, so and if you look at the extreme condition, look at the South America and Africa. So they basically the two columns are barely visible, but their sequences, their species are sequenced completely outside. Okay, so therefore, compared to everything, so there is a kind of disparity when it comes to sequencing, and you can see this the bottom chart show you the collaborations, and the white ones indicates they are without collaboration. So that means they use the gene banks, and then without consulting the native country, they did the sequencing. We can't blame them because end of the day, at least someone should uh, sequence it, right? Otherwise, what is the point having something that is not that we are not going to use? Okay, and so that is about the genomes and how the global contribution to sequencing genomes. And so once you assemble it, I already mentioned it a little bit earlier. So the second important step is the annotation. Okay, so genome annotation is very important, and you may be considering why. So, I mean, the easiest example we can take is our upcoming presidential election. So then there are candidates, right? And probably you have no idea about some so suddenly they appear. I mean, they maybe you know them, but you have to closely look at who they are. So and probably now you basically annotated 50%. Now you understand, okay, so there is a Satake. So therefore, so most probably someone Okay, so then you can look and basically annotate different regions, and then okay, so this is the organism. Okay, so this is their annotation. This is how their genes are located, where they are located, and all that information. So therefore, the annotation is very essential for a good assembly. Okay, so to uh, do this annotations, so we have different pipelines, and a pipeline is basically a workflow, and so based on your requirement, so you can basically come up with your own pipeline. And most pipelines include the information coming from the NCBI, Ensemble, and also tools like this is used to be a free tool, but it's no longer Blast to Go. And then the another tool there's another tool called Egna, and so these are mostly for the coding genes, and for the non-coding RNA. So you can use the again Ensemble, and then there are specific databases like LNC RNA database, and for the repeat elements, you can use tools like repeat marker and repeat base. And as I promised, if you're really interested in assembling genome, so there are a lot of examples. So you can extract bring files from the short reads archive in the NCBI into Galaxy platform. So Galaxy is free. You get 250 gigabytes of space, and you can do a lot of things with it. I'm conducting a couple of courses for the Canadian University. So there, I do all the bioinformatics practicals using Galaxy platform, because so my students, they don't have access to high performance computer clusters. So therefore, I mean, you can use this one very easily for your assemblies. And for me personally, as I mentioned earlier, I have the access to this data center in the Texas A&M, so which is more fast and accurate. But to use it, you need to write your own scripts. So therefore, nowadays you have ChatGPT and other clients. So therefore, it's much easier compared to where when I started those the LLMs when LLM AIs were not available, but now you can basically write your script like crude script, and then you can refine using uh, a chatbot. Okay, so I'm not against using chatbots; they are very helpful as long as you use it to improve your knowledge. Okay, so yeah, probably you guys got really bored with that big pipeline I showed you. So this is like, a very simplified version of the same pipeline. So when you receive your ODF reads or the FASTQ files, so you do 4 simple steps. That includes like quality control, because you need to check your sequence is a good quality. If not, so no point moving forward. So there are some ways to refine the bad quality ones, but if it's not recoverable, don't waste your time on it. Do another set of sequencing round. It's not that expensive, because recently we did RNA-seq for human cells. It's only $150 dollars per sample. So therefore, it is not that expensive, rather than like working with a bad data sets. And if there are like recoverable issues, you can do the trimming and filtering. So that way you can have clean rates, and then you take that clean rates and you do the sequence assembly, where you make contigs. And then, so once you make contigs, what you can do is like basically you can validate your assembly using tools like the Busco and what is the other one? Basically, you're looking at the completeness of your assembly. There are multiple tools, and then you're ready to basically go for the annotation step. Okay, so genomic is one field, and there are many supplementary fields when it comes to do our genome assembly, where we can combine not only the nucleotides, but also the proteomic data. And then, so you can basically combine the proteomic data and genomic data together and have a much better quality genome, where you have a very good annotation, and all these the gene locations, what are the coding genes, what are the non-coding genes, and what are the pseudogenes, likewise. And also remember, not only that the things you do, but also you can get more sequences from the sequence read archive in NCBI. So that way you can put everything into one pool, and then refine the outcome. Okay, so this is all. I mean, I'm going to talk about the sequencing technologies and assembly pipelines. So next, I will introduce you to like a couple of databases. So where you can get a lot of information about plant genomes, starting with the old and famous database, what we call as 1001 genome. So this is where the scientists went from sequencing one genome, that's Arabidopsis thaliana, and then they realized, okay, so there's only one. If you think about the human genome, so the first human genome, as far as I remember, there are 8 people provided their data or their DNA, and all of them were like white. So therefore, it's not giving enough diversity. So same thing with Arabidopsis, because the first Arabidopsis, so they are working with the Columbia, and then they realize, okay, so there's a lot of variation. So therefore, they decide, okay, instead of doing one, so we are going to look at a big big pool of Arabidopsis coming from different places, and then look for the variation. So that gave rise to the 1001 genome. And end of the day, their objective was to do 1001 genomes, but they did around 1300 genomes when they published their paper. Okay, so and then another database is called the 10KP, and so this is it's still ongoing genome initiative, genome sequencing initiative. So what they're doing is basically they want to sequence, and it was 1,000, right? 10,000 plant genomes to identify different genome architectures in plants, and it is initiated by the Chinese uh, Science Foundation. I think it's Chinese Science Foundation. And so they are basically it's contributed by China National Gene Bank. So they are basically, if you're interested, you can also send your genome sequence to them and have a collaboration. Okay. And if you're really interested in crops, the rice, So they basically sequence 3,000 rice genomes, and you can access the 3,000 rice genomes from Rice Snake database in the International Rice Research Institute. So there are 47 rice species coming from Sri Lanka. It's there, and then many other thousands of species coming from other countries. And so therefore, I study rice. So therefore, every time I want to see the variation between different cultivars, I use this database for that purpose. And if you work with other plant genomes, including the ones I already discussed, the ensemble is a very good resource for you to use. It is very well curated. For me, I find it even better than the NCBI. So there, you have all the genome information. You can find genes, transcripts, proteins, and also you can bulk download sequences and gene and all the information using this tool called Biomart. It's inbuilt into this system. Okay, as you can see here, there are 56 release, and it has thousands of different genomes. And there's uh, kind of a sub part of this genome. It's what we call as the Grammy database, where you have crop genomes. And so if you work with rice or not Arab, it's not crop, but it is still there, like wheat. So this is a very, very nice curated data source hosted by the Cold Spring Harbor Lab. So where you can use, and also this database is similar to the ensemble JGI phytosome. It also has many genomes available in this database where you can easily access, download, and use for your own studies. Okay, so another database. This is basically initiated by the Japanese government, where it's called Plant Garden. It includes genome and resource database entry, where you not only you have genome, but also it contains marker information, especially if you're doing breeding. And if you're a crop scientist, this is a very, very good data source. Okay. And then another database I would like to introduce is Earth Biogenome. So the objective of Earth Biogenome is basically sequence every species in this world. Okay, so that is their initiative, and it's based on Arizona University. And so they are planning to do sequence all species. And so this shows you the current progress. And please visit their site, and then you will find a lot of cool information regarding the genomes they have. Okay, so hopefully at one day they will finish their task. Okay, so now we are wondering, okay, so we talk about almost 1.5 hours about the genomes. What is the use of it? Okay, so we can use the knowledge we gathered from this genome information to do many things. Okay, so I know most of you are really interested in the biodiversity. Okay, so and we always blame especially modern the agriculture or the classical agriculture, so that we basically ruin the diversity. Okay, so one example is if you think about the tomato, so there are very few variants of tomato you find in the market. Okay, so scientists study the tomatoes from the South America, the good old ones. Those are not actually edible, and then the current ones we have, they look at the changes using genome information. What they found out was there are not really a lot of gene level changes, but instead there are changes in the promoter. Okay, so non-coding region. So that means, so they realize if we reintroduce the promoter changes, we can have a vast range of trait variations in tomato. See, So you can bring back the lost diversity using not even introducing the genes from different species, no transgenic, but rather using a genome editing, you can make changes in the promoter region and then bring back the lost glory. Okay, and also you can identify the genes those are responsible for high yield, high productivity, by comparing highly productive variety versus let's say uh, average variety, and then you can basically do the necessary changes and then increase the yield of it. Because remember, if you want to save the environment, so you need to make sure that the unit productivity of agriculture land is high. Otherwise, what is going to happen? Let's say you go for organic agriculture. Organic agriculture, usually your yield is less. Although you sell it for a huge chunk of money, the yield is less. So now what you need, because the population is increasing rapidly. Think about it. Now, you need to feed these people. So that means you need to make more food. So what is going to happen now? You are trying to basically feed them with organic food, like the government tried to do it in 2018, right? The stupid 2020, the stupid plan there. So now you need twice more land. So then you need to encroach the forest area. Okay, so you think you're doing a good thing, but in reality, you're trying to destroy your forest, because so they need to be fed. Otherwise, they are going to revolt. I mean, the start revolution. So therefore, I mean, that is the situation. So you need to think logically. We need to use all technologies, all this genetic knowledge, and all the tools in order to increase the unit productivity. So that means we can have more lands for the forest. Okay, and also we can use this information in order to make future-proof crops. So that means the crops that can stand in very harsh conditions and still give a good output or good yield. Okay, so I have one final slide where I would like to give you the latest and kind of greatest advantage of genome science. So that was if you think about the corn. So I don't know whether you already learned about it. The old world corn is the teosinte is this size, and it has if you look at the kernels, I mean you can't eat. So basically you need a hammer to break it open and then basically consume it. And that was what our ancestors you found in the good old days. And now we have very sweet corn cob, right? It took 10,000 years for our ancestor to genetically engineer that barely edible teosinte into the modern corn. Okay, so but there's one mystery, because the corn was originated in the Mexico area, and it's really hot. It's not basically adapted to the cold climate. Okay, so scientists were thinking, okay, so how this happened, how it basically spread it all over the North America. And very recently, when they did sequencing, the genome sequencing, they found out. So good old days, there was a cross between two teosinte. One that was growing in hills, that which is more tolerant to cold, and then the whatever the hot the teosinte. So that cross helped the corn to spread all over the North America, thereafter all around the world. Okay, so this is a mystery that was unknown for the human for nearly 5,000 years. Okay, so see the beauty the genome science can bring to our hands. Okay, so with that, I would like to finish the lecture, and I'm more than happy to answer any questions you have. And I hope I didn't I hope you guys are still awake. Okay, thank you.
Thank you, Professor Herath. It was really inspiring and very informative lecture. So I really enjoyed every minute of the lecture. So I didn't just do anything. I just listened to your talk at a stretch. I think the students, so that is actually I wanted to give to students, right? So to stimulate their enthusiasm on the beauty of the genomics. So I know you all in your 4th year, so you will go for your higher studies in near future. So try to explore the genomics. The beauty of genomics is there. So the example is here. So you have inspired by Professor Herat's lecture. And so I'm really happy, and also I'm giving my fullest dedication, and also thanks so much for your wonderful lecture